second session on instrumentation and nanophotonics. I think to present their innovation on multiplexed fluorescence sensing in a small form factor device.
Thank you for that introduction. Good morning, everyone. Thanks for taking the time to be here. My name is Liko, and this is Gagan Minhas, and we're going to talk to you today about our fourth-year design project in partnership with Spartan Bioscience. As you mentioned, it's a four-fluorophore imaging measurement system. So an understanding of genetics is a very important tool for modern medicine. Your genetic makeup influences every facet of your body's function, whether it be your hair color or your disease propensity. Of course, all the most interesting genetics differ between individuals, which means to take full advantage of the information, every person needs to be tested. Spartan Bioscience has in production right now an on-demand individual test for the CYP2C19 gene. And that gene just affects how your body metabolizes certain heart medications. They achieve this testing through a 2-fluorophore imaging system and PCR for DNA augmentation. Now, Spartan solution is, I won't say revolutionary, but it's very important because it helps doctors who originally had to prescribe this type of medication on a trial and error basis. And I don't know about you guys, but that's not how I want to be prescribed heart medication. Spartan is seeking to expand their technology to unlock even more of the power of genetics. And to do this, they've determined that they require moving from their 2-fluorophore system to a 4-fluorophore system. So in the hopes of contributing to this field in a big way, we decided to partner with Spartan and tackle this problem as our fourth-year design project. Our goals can be broken down to the following. We want to be able to track an increase in active fluorophore concentration of the 4-fluorophores, and these concentrations can be quite low. Spartan's determined that they can be down to 0.01 micromolar. As a secondary requirement, we want to be able to track an absolute concentration as opposed to the relative change, which is what Spartan really requires. And if we do reach that goal, we'd like to be 5%, within 5% error of the actual value. We'd also like to keep our size relatively small, ideally within about half of a shoebox size, just to get a visual of it. We also want to maintain compatibility with Spartan's current solution. That means using the 2-fluorophores they use now, and also using the custom test tube which they've developed for their system. Finally, we want to keep our cost low. These types of systems can run tens of thousands of dollars. To be competitive, we'd like to be at or under $1,000. We also don't have tens of thousands of dollars to spend. So genetic testing has been of interest for a few years now, and a lot of the focus is shifting to make it more accessible and more applied in a useful way. Spartan's RX system is excellent in terms of accessibility. It's quite small. It's actually about the size of a shoebox, and it's very easy to use. And there are competitive systems like the Biorad CFX system, which also use a 2-color process. And the problem with these 2-color processes is they're not as versatile. On the other side of things, there's lab-focused systems such as the Cepheid Smart Cycle, which can run multiple samples at once and therefore test multiple genes at once. The catch with these lab-focused systems is that each channel you want to look at requires a different sample. So to our knowledge, there's not a good system that combines the small form factor and versatility of a single sample 4-fluorophore system like we're looking at. So the number one challenge in meeting our goal is going to be the idea of multiplexing, finding a reliable way to track the change in each fluorophore individually. We see two major hurdles to reaching this. The first is that we want to track 4 fluorophores independently, but any camera is only going to give you 3 independent channels. So unless we get really creative, we're going to have to deal with some mixing on any given channel. The other side of things is fluorescence energy transfer, where the emission from one fluorophore is absorbed by another. This artificially reduces our signal, and having the 4 fluorophores in the tube puts us at a high risk for this. Another challenge we expect to face is the low concentrations, as I mentioned before. Spartan predicts having these low concentrations. We need to be able to detect them. Our camera noise level and its sensitivity is going to affect this, as well as the environment we're working in. So it's a consideration for us. Yet another challenge is, as I mentioned, the cost. So clearly from Spartan's experience and from competitive products, these aren't cheap devices. We expect a pretty high cost to come from the specialized optics equipment. So given our limited budget, that's something we're going to have to look at. Now, the final challenge is one that I'd like to say we predicted beforehand, but unfortunately we didn't, and that has to do with replicating the actual use case of the device. So in an actual system, the test tube is held stationary while PCR is performed, and images are taken throughout. This allows you to compare the image brightness back to an earlier baseline value and see if you're getting a rise, which indicates successful PCR. In our system, in our test system, since we're not actually running PCR, we need to simulate this. The way to do that is obviously to take different samples of different concentrations and put them in the tube one at a time and compare them that way. The catch there is that each tube is not quite equal, whether it be scratches or dust on the tube, which changes our brightness slightly, which in turn makes our trend a little bit harder to elucidate. So the first step we took to mitigate the challenges that I mentioned was careful selection of fluorophores to minimize the absorption spectrum overlap. This allows us to activate them essentially isolated. If you look at the absorption profiles there, you can see that it's really only the tails of each one that are overlapping, so we can essentially target one at a time. The other side of that is minimizing the overlap between the emission of one fluorophore and the absorption of another, and that's to minimize the fluorescence resonance transfer, which I mentioned before. Also, in order to help eliminate the multiplexing issues, we performed a really thorough calibration that allows us to work backwards from the brightness value, which is our measurement, to a concentration, and it also allows us to predict any overlap we might see. We also specified an analysis process which gives us more control over this kind of overlap. By working from the lowest energy fluorophores to the highest, we can keep track of the concentration of each fluorophore as we go, which helps us to account for the mixing as we get into using the higher energy LEDs. So in order to keep the device simple and as low cost as possible, we decided to offload processing to a host computer. This actually has the added bonus of making the device a little more versatile. You can plug it into any computer that has the software installed and have it run. So the physical design of the system is actually quite straightforward. First, we designed a custom tube holder to fit Spartan's custom tube. And I will apologize for the black on black picture there, but you can see where the wires all go. That's our system. If you come by the booth, you can see it. All around the tube, we put the 4 LEDs to shine in. And the way the enclosure is designed is to minimize any stray light from getting in and affecting our signal. We then mounted the camera beneath the tube because the bottom of the tube is actually the only flat surface, so it's the best for our imaging. And then we put the whole thing in a blackout box, again, to eliminate stray light. In an actual PCR device, it would probably be contained in a metal enclosure. So the choice of fluorophores and illumination was a little bit more precise. We chose our fluorophores from Spartan's current supplier to make ordering a little easier, and we went through their entire catalog to find the minimal overlap we could, as I mentioned before. From there, we were able to look at the LEDs and target our LEDs to the specific absorption peak of each fluorophore. That allows us to achieve what I mentioned earlier, where we would only illuminate or only activate one fluorophore at a time. Before we actually ordered, we went through a very in-depth simulation process to ensure that the LEDs and fluorophores we chose would actually give good differentiable results with the camera that we chose, and we found that it did. And this simulation actually helped us in developing our analysis and our testing process as well. To interface with the camera, we only needed a USB connection to our host computer. To interface with the LEDs, rather than designing our own circuit and drivers, we just went with an Arduino for the time being. It's convenient in terms of USB connectivity, it's easy to program, and it allows us to control the brightness of our LEDs, which was important because that was something that we had to work through in the process. So the testing portion of our design was actually simplified a great deal by our careful selection of fluorophores and LEDs. So the first stage of the testing involved building calibration curves for each fluorophore. The way we did that was going through isolated concentrations of each fluorophore and activating them with their target LED. And then we would take the images that we obtained from that, break them down into the red, green, and blue components, and look for trends on each channel. And whether it was 1 or 2 channels, we saw the best trend. That's what we would use in future to go back and correlate future data. So with the calibration curves done, the next stage is, in the testing process, requires that multiplexing samples be handled. So the way to do that is to just create a series of varied mixed fluorophore samples and run them through our testing process to see that we can detect a change in one or more of the fluorophores. So now that I've gone through and talked about our design, I'm going to pass it off to Gagan, who will explain what actually happened.
All right, thank you, Nick. So I'll begin by talking about the design challenges that we faced once we began imaging, taking pictures of our samples, and basically doing the analysis. So the first of two major complications we encountered was how our camera responded to the emission of BioSearch Blue, our UV-excited blue-emitting fluorophore. Now, being a small volume fluorophore, our provider was unable to provide detailed specifications which would have made it easier to prepare for. Regardless, when we went to test the desired concentrations of BioSearch Blue, we weren't able to actually register a significant signal for the area of interest on the image relative to the background of the image, irrespective of the sample concentration. Now, our first approach in solving this issue was to increase the UV LED brightness and the camera exposure time to their maximum setting. Now, this did in fact increase the signal that we were able to measure, but it had the added effect of increasing the background noise level. So under these conditions, we weren't able to actually build a strong correlation between sample concentration and image brightness. Temporarily, it was determined that increasing the concentration of BioSearch Blue that we were looking at would be the optimal solution. Now, BioSearch Blue is not one of the fluorophores currently in use at Spartan Bioscience, and after consultation with them, it was agreed upon that this would be an acceptable solution. Now, thankfully, in later discussions with our camera supplier, we were able to determine that our minimal response was in fact due to a calibration error associated with the camera, and we were able to return our concentration range back to the initial range that we had selected alongside our other three fluorophores. Now, a second unexpected problem arose from our somewhat unrealistic method of verification that Nick mentioned earlier. As we said before, in an actual real-time PCR device, the unquenched fluorophore concentration increases with time due to the thermal cycling, which augments the DNA. Now, our system is not yet integrated into an actual PCR device. So as Nick mentioned before, we need to simulate this condition. The best way to do this is to have individual test tubes where we vary the concentration of the fluorophore that we're looking at. Now, this had the unexpected issue of us examining the individual test tubes and finding that depending on which tube you were using, you had scratches or apparent bubbles that would come in the actual tubes, and as such, there would be a change in the apparent brightness of an image. Now, in the actual system, this isn't so much of an issue. It's actually pretty much expected and accepted as an issue. However, in creating an empirical relationship between concentration and brightness, it resulted in some difficulty in our initial testing. Now, to verify that this was actually the problem and not some sort of problem in regards to our actual analysis technique, we needed to perform a second test. Now, this second test involved us re-rigging our entire system to accept the standard cuvette that you would use in, for example, UV-Vis experiments. We would then dilute the sample directly into the same test tube to make sure that the variation due to the scratches or dust found on the test tube, as well as variations due to tube alignment, was kept constant, as you would kind of do in the actual PCR system. With these results, we were able to show a much more consistent trend and allowed us to conclude that the greatest source of error was, in fact, the different test tubes that we were using. Now, in the end, the solution to this problem will be statistical. Our goal is to collect as many images as possible to develop a good, usable trend. So the important part, of course, are our results. For our device, as mentioned before, it's important to perform a significant amount of testing to accumulate a good set of baseline images and to further establish the distribution. As such, verification is still ongoing. However, from the graphs you can see above, we do have results that we can share with you. First is the calibration curve for each of the four fluorophores. 6-FAM, our green, BioSearch Blue is the blue fluorophore, Quasar 705 is the red, and CalFluor is the yellow. These graphs show that we were able to detect the fluorophores individually and how they behaved in isolation once you varied the concentration. Now, as you can see, the graphs are more or less sensitive which depends on a variety of physical properties between the LEDs, the camera, and the solution that we're currently working with. But the great thing is that there's a strong correlation, which is linear, that we see the minimum R-squared value being at around 0.95, which would be for the Quasar 705. Now, the next frontier is multiplexing, which, as Nick mentioned already, is the mixing of multiple fluorophores in the same sample at varying concentrations. To this point, we've tested 5 different multiplex samples, 3 of which contain an even mix of all 4 fluorophores, and 2 of which contain pairs of fluorophores, ones that we thought would basically give us the most trouble analyzing, and these are our red and orange tags and blue and green tags. Unfortunately, as I've mentioned before, because of the recalibration that was required with the camera, we are still working on collecting further images before performing analysis. So far, our results are good, but we aren't meeting the secondary requirement for a 5% determination of the absolute concentration. Based on the systemic limitations that we're faced with, we don't believe that this goal will be reached by the end of term. However, we have definitely reached our size goal. You can all come later on today to take a look at our booth and view the prototype that we have and a demo of it as well. We are easily meeting the half shoebox size constraint, as could be told by the earlier image that we had. We've also maintained the level of compatibility that we'd hoped to. We are using Spartan's test tubes in our setup directly, and we are employing 2 of the fluorophores that they currently have in their day-to-day operations. Regarding cost, we have been remarkably successful. Our camera system and the lens basically rounded off to our greatest cost, which was around $350. At $100, we were able to purchase our LEDs and the Arduino board, and the mechanical components alongside the test tube holder rounded off for about $50. Given that a second stage version of our system would need to integrate with PCR, the test tube holder would need to be changed from plastic, ideally to aluminum, to handle the thermal cycling. And we believe that a replicate of our current design would cost somewhere around $600, which is well under the $1,400 budget that we've been given. Now, there are a few steps which are still being taken before wrapping up the first iteration of the design, namely collecting a large number of multiplex sample images and performing the subsequent analysis. Additionally, automation of the testing process is always a good idea, and we believe we are at a safe point in the design to approach this idea. We are also at a good point in the design process to talk about our second iteration. This would focus on the analysis algorithm by incorporating a better error detection and error checking methods to handle the test tube variation that I talked about earlier. Also, from a hardware perspective, bypassing the Arduino board would make the system much more cost-effective, less complex, and increase durability. An LED driver would need to be designed, which would interface with the host USB port and would accept brightness control. Designing a custom circuit board for the LEDs would make the system more electrically robust and durable by removing loose wires as well as the breadboard circuitry. Also, the next iteration should make itself compatible with real-time PCR. This requires a basic restructuring of our mechanical system as it is to accept thermal cycling via heating and cooling, and we have brainstormed some possible geometries that would allow us to handle this issue. We also need to change the tube holder material to one that is heat conductive and much more durable. Performing real-time PCR, lastly, would eliminate the tube exchange issue that we are currently facing, which in our case is an added benefit. That is currently where our design stands right now. It's been an exciting process with its shares of ups and downs, but I'm glad to say that the end is near. Thank you for taking the time to listen to our presentation, and we'd like to open the floor for questions.
So we have 2 minutes for questions. Chris?
Okay, so good work. So as far as I understand, you've got the ability to say yes, no on any of your channels.
Right.
Next step is being more quantitative.
Right.
There you're going to have difficulty distinguishing, is this a strong green or a weak blue? Because of bleed over effects. So how are you going to handle that in, 30 seconds or less?
Right. So the way we expect we're going to handle that is we're only activating one fluorophore at a time. So if I can go back to the spectra.
When you turn on the LED, you're probably going to get a response from any one of the nearby fluorophores. You can't distinguish unless you play some interesting tricks.
We've actually thought about this, and we have, well, in my opinion, 2 kind of methods that we could approach. The first is a very empirical-based approach, and that is to basically know that when we turn on the green LED, from the spectra that we've been able to establish, we know that we're going to turn on 6-FAM, which is our green fluorescent molecule. Now, because intensities are additive in the system, we're then working to basically develop a calibration curve that says, let's figure out what's the concentration of 6-FAM. If we can correlate that to the brightness, since brightness is an additive property, we could theoretically subtract it out when looking at the blue channel to see how much biosurge blue is actually present. And hopefully, if that biosurge blue lands on our calibration curve, it would be detectable.
Okay, good stuff. That's good. I like that. Thank you.
That was 20 minutes, okay. If there's like a very short question with a short answer, then we can take it if you want.
Just say it.
Otherwise, who knows what happens?
All right, so let's thank our presenters once again.